Okay, am I audible right now? Everyone, please give me a thumbs up if you can hear me. Okay, let's just wait couple of minutes for others also to join for the session. Right now we have five people here with us. So let's just wait a few minutes for the others to join for the session. Meanwhile, you can share this with your friends or your family member who are just interested for the STEM activity. Okay, I think we can start the session. So, welcome everyone for our today's YouTube live session with 1090. I am Alma, STEM Educator, and I'm super excited to have a session with you all today. So, today what we are going to do is we are going to extract DNA from banana. Yes, you heard it right. We are going to extract the real DNA from banana, and you are able to see it. It will be visible to you. So we know that DNA is present almost in all living organism, whether it is fruit, plant, or human or animals. It is present in all living cells. So what we'll be going to do is we are going to understand some things in the session. So first, what we are going to understand is what is DNA and how where you will find it. Second, what we are going to understand is how we can extract the DNA using just kitchen items at home only. You don't need fancy lab to extract the DNA. So this is what we are going to learn. And next thing what we are going to learn is how we can spark the curiosity among students in classroom by inculcating this activity into our lessons. So I think we are good to go. Before starting the activity, I would like to ask you all one question. So the question is, which fruit do you think has more DNA? A banana or strawberry? Which fruit do you think has more DNA? A banana or strawberry? Please comment down in the chat below so that I can see your responses. Okay, I can see strawberry. Other responses, which fruit do you think will have more DNA? Strawberry or banana? Waiting for few more responses. Banana, okay. Strawberry, two strawberry, two banana, okay. Banana. Mm. Can we have few more responses? Banana. Okay, I can see some are saying banana, some are saying strawberry. So we'll get back to this question at the end of the session to get the right answer. So stay here with me till the end of the session so that you can know the answer. Uh, Wait a moment. Let me just start the activity. Okay, please give me a thumbs up if if you are able to see the setup. Please give me a thumbs up if you can see the setup. Okay, thank you for the responses. Before we start the activity, I want you all to keep the alcohol or sanitizer in the freezer right now only, because we need it in chilled form. So it will be good if you keep it right now only in the freezer. So by when we'll need it, it will be there with children. So first, we'll do the first step here, in which we need a ziplock and a ripe banana. So what we are going to do is, we are going to peel the banana. Okay, just peel the banana and then add this, or put this into a ziplock. Close it. Once it's closed, just mash the banana. Those who are doing this activity with me, I guess they must be enjoying doing this, since it is so satisfying to mash the banana like this. So yeah, just make sure it's all mashed properly. You can even use spoon here to mash it completely. Make sure there is no lump present in the banana. It's fully mashed. So let me tell you why this step is necessary. Basically, we are just breaking the cells of the banana so that we can take out the DNA from it. So this step is breaking the cells. Take few more seconds to completely mash it. Okay, I think we are good to go. Let's keep this aside for now. Okay, let's keep this aside. Now we'll move to our next step. So for next step, what we are going to need is a half glass of water, 1 cup of water you can use. So in water, what we'll add is 1 teaspoon of salt, we are going to add into this. And then 1 teaspoon of dish soap. Okay, if you don't have dish soap, that's okay. You must be having detergent powder at home. Instead of using dish soap, you can use detergent powder as well. And what we are going to do is just stir it slowly. Do not stir it too fast because then bubbles will form and we don't want the bubbles to form into this. So just stir it slowly so that it is formed into a mixture. So this is basically we are making a DNA extraction solution here. By the time I'm stirring it, take few seconds to think about what will be the role of salt and dish soap into this, like what it will do to the banana if I add it into the banana. Take few seconds. Okay, I think it's done. What we are going to do is in the ziploc we have mashed banana, right? Or if you have mashed your banana in a bowl, what you need to do is add this DNA extraction solution into the mashed banana. You can add it so here I have used around 7 to 8 teaspoons, I guess. So you can add it according to you, like if you want more DNA solution you can add more, if you want less amount of it then you can add less. Yes, I have added more than 10 spoons. Okay, now what we are going to do is we are going to lock this again. Okay. Now just mix this solution properly with each other. Do it slowly so that not more bubbles form into this. Okay, I have asked you one question, like what is the function of using salt and dish soap over here. If you can guess, you can write the answer in the chat box. So basically we need to wait like for few minutes to make this mix properly. So let me tell you why we are using salt and dish soap. So dish soap is necessary basically to break the cell membrane of the nucleus so that the DNA comes out uh, from the cell. So that is why we are using dish soap, whereas we have used salt over here because uh, to neutralize the DNA charge. So DNA is negatively charged, So basically, adding salt will help us to stick the DNA strand to each other properly. So there is less repulsion between the strands of DNA. So these are the function. Okay. So what we'll do is right now, next step is we are going to filter out this solution. So what we'll need is a glass and a strainer. Okay, so this is a normal tea strainer which I am using right now. If you don't have a tea strainer, you can use tissue paper or a cotton cloth to make the filtration. So what I'm going to do is I'm going to add this solution. So here you can see so here we need to filter this out. So whatever is dripping down the glass is our DNA solution. Basically a solution which will have our DNA in it. This will take few minutes to filter it. By the time it is getting filtered, I have one more question for you all to think about and share your thoughts in the comment section. So how can you use this activity in your classroom, or how can you inculcate this activity into your lesson so that it will create curiosity among your students? You can share your thoughts about it, like in what topic this activity can get included into your lesson. Okay, I can see some responses like heredity. Yeah, you can for sure include this activity in the topic like heredity, where to let know students what are DNA and how DNA has some information which passes from one generation to another generation, which is basically responsible for transfer of characteristic. So here you can see I have a solution. This is my DNA solution. So we are in the final step of the DNA extraction, which is the most exciting part, I guess. What we are going to use is we are going to add uh, alcohol or hand sanitizer into this solution. So just pour alcohol in the same quantity of our DNA extracted solution. So you just need to tilt it tilt this a bit and I'm adding here sanitizer. Make sure it is in equal proportion, 1:1 proportion. The okay, just pour it slowly, do not stir it, and just observe it. Let's take few or couple of minutes to observe whatever you are observing right now. Write down in the chat box. The quantity of the sanitizer should be equal to the quantity of the extracted solution which you got after filtration. You can just share what you are saying right now after observation. Do not stir this, just keep it still. If you observe it clearly, you can see some thread like structure is forming or white substance is forming at this surface level of the solution. Let's wait few more minutes. Okay, so I have a question over here. Why do you think after adding alcohol, the there is a separation of the solution into like white layer and the banana solution? So why there is a separation after adding the alcohol into it? Why it's happening? What do you think? You can answer it in the chat box. Basically, the separation of into two will be in a scientific term will be called as precipitation. So why this precipitation happened after adding alcohol? If you have the answer, please type in the chat. Okay, so basically, adding alcohol has made the separation because DNA is present inside the cell where there is a water around itself. So it is so basically it is used to water and it can dissolve well in water. So after adding alcohol, it becomes insoluble because DNA is insoluble in alcohol. So that is why you can see the separation or the white layer at the surface level of the solution. So what you will have to do right now is use a toothpick or a bamboo stick here, and we are going to slowly swirl it, not too hard, just slowly, so that the white thing can you know so you can just use a spoon to take out the Okay, so we are able to extract this much of. So those who are able to extract, they can see a clumsy stringy thing at the upper level which was present, which we were able to extract it. So right now we can say we have a coat in our hand which has made the banana like a banana. Okay, give me a moment.
पहले मेरे को लगा पहले ही बता देंगी.
Okay, yeah, so we were able to extract this much of uh, DNA from it. So I hope you have enjoyed this session. I have a question here. Let's get back to the question which I have asked at the start of the session. Like, if we are, which fruit will have more DNA, strawberry or a banana? So, lot of people commented strawberry, and some of you were said banana. So, without revealing the answer, I'll just explain or just share an information. From that information you will be able to come up with an answer. So basically, banana are triploid, meaning they have 3 copies of chromosome in their cell. And if I talk about strawberry, they are octoploid, meaning they have 8 copies of chromosome in the cell. So you can just think about banana being 3 copies of chromosome, and strawberry has 8 copies of chromosome. What do you think? Like, which fruit will give more DNA? You can comment down in the chat box. Strawberry. Okay, Nida. Thank you for the response. Anybody else? Thank you, Shami Miss. Okay. So yes, strawberry is the correct answer, because the more the copies of chromosome, the more will be the DNA. Yes, so chromosome is basically which is like a spindle of DNA strand, which the chromosome is made up of that. So as humans have a pair of chromosome like that, strawberries has 8 copies of chromosome. So in that way, strawberry will produce more DNA. So that is the correct answer. Uh, Basically today what we have learned is how to extract the DNA at home without any fancy lab or without using any lab equipments. We have extracted real DNA. Here I have actually I have one more question, not for you to share right now, and one challenge for you to do it at your own time. So the question is, you can see in textbook or like while understanding about DNA or learning DNA, we must have seen that DNA can only be seen through microscope, but here we can see the DNA through our naked eyes. So how it is possible? Why through our naked eyes we were able to see this extracted DNA? What difference it makes? So you can reflect upon this question and research it and get came up with answer. Second thing, the challenge is we have used here banana for the extraction of DNA. Can we use the same step for any other fruit or any other vegetable? Uh, for example, apple. Can we do this with apple? So this is your challenge uh, for today. So I have one more question for you all to answer. Why we have used banana in this activity? What do you think? Like, why I have used banana over strawberry? I could have used strawberry here because strawberry is giving me more DNA extraction. So why I choose banana over strawberry here? Any other responses, whatever you feel like? Any reason for choosing banana over strawberry? Okay, because it is easy to mash. Yes, obviously it is easy to mash. It is soft. It's clear. Any other thing? Okay, so I have preferred banana over strawberry here because banana is easy to buy, yes, Nida, easily available at home, or you can buy it at any season at low cost. Whereas strawberry is very seasonal and it will be costly, and to preserve it you need refrigerator to preserve it, and there are seeds in strawberry which will come as a barrier while we are filtrating the solution. So that is why we can prefer banana doing this activity in classroom or at home. So yeah, we have come to the end of this session. uh, I have enjoyed a lot the session with you all today. I hope you have also enjoyed it. Do share your result with us by tagging us like hashtag 1090. Do like and share this video with your friends or family members, and do not forget to subscribe our YouTube channel 1090 to get an update for more such fun activity and videos. We'll meet you again next time, same day, same timing. So see you there. Bye. Thank you for joining.